understand the good side of bacteria, stay tuned. Well, Ashley Franks reveals all.
Thank you. So I work with bacteria. People always think of bacteria as being bad, but less than 1% of bacteria are actually detrimental to us. So most bacteria are actually quite beneficial. We need bacteria. Bacteria are one of the fundamental things on Earth. Without us, if humans disappeared, Earth would be fine. If bacteria disappeared completely from this Earth, we would have a very short time before everything collapses. But some of the ones that I'm going to talk about today that's interesting to us is bacteria that can eat and breathe electricity. So this is very important for a lot of beneficial processes. Some people might find this a bit shocking, but I'll talk to you about some of these ideas and some of the things that we're working on. But when we talk about electricity and bacteria, basically it all comes back to food. Bacteria are like us. Food is what drives them the most. Some other people might think there are more important drivers, but try and think about that when you're hungry, and you'll soon find out that food's the most important thing for us. But when we take food in, what we're doing is changing the energy in that food into something that we can use ourselves. So when you eat too much of some food, you store that energy and we all get a bit fat. If you want to lose that fat, you have to use that energy and convert it into something else. But this is all done on our cellular level. Here, all it's showing is that what we're doing is breaking down all those chemical bonds in something like glucose. We get the energy because of the electrons from there. It gets transferred to oxygen. We breathe in oxygen. And what actually occurs in this is that we're actually now converting energy from a high state to a low state, and we take a tax in between by forming this ATP and these other chemical reactions that go on. So this one's quite important. Interesting one to remember. We breathe in oxygen and we breathe out CO2 and water because of all this reaction. Tonight, when you go to bed, weigh yourself before you actually go to bed, weigh yourself when you get up, and you'll notice there's a little bit of energy loss because through the night, you've used up some of your glucose stores, changed into CO2 and oxygen and breathed it back out. But bacteria do this exactly the same process. Same thing, it's just about following the reaction, following the electrons. The electrons come in, they actually get passed through this sort of membranes, these proteins to make this ATP, but in the end, what the electrons get passed to is oxygen. So this is what we know. When we breathe, we need the electron acceptor. Without an electron acceptor, any oxygen, then we don't do very well. There are a lot of bacteria that can breathe oxygen as well. They go through the exact same process. And you may be wondering, well, what has this got to do with electric bacteria? And the thing with bacteria is that they're a lot more creative than us. They've been on this earth a lot longer than us, and they can breathe a range of other things than just oxygen. So they can breathe things like nitrogen, different sulfur. But the things that we're really interested in is that the bacteria can also breathe iron. So same process. Instead of glucose, this is just acetate, a chemical reaction. But what the bacteria are doing is giving their electrons to these lumps of iron instead of oxygen. The interesting thing here, though, is that this is not a gas. It's a solid lump of metal. So the bacteria are able to go through their processes. Instead of breathing oxygen and doing that internally, they get their electrons and transfer it externally. At the same time, you get iron gets reduced. It's the electron acceptor. Instead of water, you get iron too. This one can be made to be fun because when you have iron oxide, here it's just mud, whereas you can actually change it with the bacteria respiration to magnetite. And that's then magnetic. So when you're going to Mars, when you're going to these other planets, this is the type of thing they're looking at for these microbial signatures for this type of biological procedure. But the thing here is, of course, the bacteria can actually go through and transfer electrons outside themselves. They can carry out a number of useful and interesting sort of processes. And one of these is that it's not just iron that they breathe. They can breathe a range of different things, such as uranium. So uranium is just another metal. Uranium-4 here, when it's glowing, it can be used as an electron acceptor. The bacteria can transfer an electron to it. But the good thing in this process is uranium-6, it is dissolvable in water. It's dangerous because it can dissolve in water. Uranium-4, still dangerous, but it precipitates out. So if you actually have bacteria that can breathe this uranium, and this is what's being used at some sites in America, is that the bacteria, here it's an old part of the Manhattan Project from World War II. Used to be a whole bunch of uranium being done here. There's uranium contamination. That's going into the Colorado River. You don't want to actually be contaminating your local water supplies with uranium, especially when that river is the one that flows through the center of America. But what this allows you to do is, if you can understand this process, is you can feed these bacteria here acetate, which is vinegar. If they actually go through and breathe that uranium, transferring their electrons to it, what can actually occur then is that that uranium will precipitate out, sit in this soil, and actually not go directly into that river. So just using these bacteria's ability to breathe electricity, you can actually prevent a lot of contamination going on. But most people actually associate electricity with something like a battery. And what a battery does is just carries out a chemical reaction as well. It actually gets electrons from one place to another, and this is what's been happening with these bacteria in this uranium and these iron reduction sites. So we actually do have ways where we can actually use an electrode instead of the heavy metal, oh sorry, not the heavy metal, but there's the iron or the other metal, where the bacteria can actually, through its breathing places, can turn acetate into electrons that we can go through and collect. So this is a natural part of breathing. If this was us, that electron acceptor would be oxygen. But these bacteria can now use a graphite electrode as their electron acceptor. So this will be similar to me if I went and breathed by touching the wall. So these bacteria are just breathing, but instead of taking oxygen in, what they're doing is using the electrode here as their electron acceptor. This is like their oxygen or their iron. So this at first seemed like it was quite interesting and going to be just specialised in what bacteria can do it. But if you actually go and put these type of simple devices in, it's like a battery now. You have an anode and a cathode, your positive and your negative, electrons flow from one to the other. You can actually bury this in mud anywhere in the world, and you'll end up finding that the bacteria there that can actually go through and produce this electricity. So what we have here is just a very simple device. It's just an anode and a cathode and a resistor. It can actually be buried in mud, and because of all the bacterial reactions that's going on there, the bacteria are breathing and they're actually producing this electricity that you can collect as a small electrical current. This can be quite useful because even though it's a low level of power, with microelectronics now, if you're in an area that you're away from sunlight, if you're in an area that you can't actually easily get and change batteries, then these types of things can actually give you can give you a steady supply of electricity over a very long period of time. And the US Navy has been interested. This is a larger one, but this can give the equivalent of about 49 D-cell batteries per year. It may not seem like a lot, but if you have to go down the Mariana Trench to change batteries, it's actually a better way to do it and actually save on those changes in swimming in and out. But this discovery showed that we could isolate bacteria into pure culture. So these are single bacteria now that we can make these microbial fuel cells out of, and we've got these new bacteria that we call electricogens. Technical terminology is that electricogen is a microorganism that can completely oxidize organic compounds to carbon dioxide with an electrode serving as the sole electron acceptor. This is what we do with oxygen. These bacteria can do with just an electrode. And they can grow on a big wide range of different surfaces, anything that can actually collect electrical current from. These bacteria we can actually go through and actually grow and study and look at their interactions. And this is all back then to the acetate being used as their electron donor and using an electrode as an electron acceptor. Because in all these ones I've been talking about, it's just taking about electrons from one point to another. But the interesting thing that was being looked at with a lot of these bacteria is we eat food to get the electrons and we breathe oxygen to get rid of the electrons, getting that redox balance in between. These bacteria, some of them, you can actually reverse this process and actually feed them the electrons and get them to actually breathe something else. So like I was saying, that bacteria can use a big wide range of other electron acceptors other than oxygen. Same thing here, as long as you're given the electrons at the right electrode potential, these bacteria can eat those electrons, pass them to the electron acceptor, and get that difference in the redox power. And this is actually quite useful because in a lot of sites, the problem is here is like the uranium contamination or there's a chlorine contamination, is that the bacteria are there that can break these down, but they just don't actually have the food available. And if you went through and dumped a whole bunch of molasses, or if you dumped a whole bunch of something in, you know what happens when you leave food out, your compost heap, lots of bacteria will go crazy for it and there'll be big competition. But what we can do with these type of ones is we can feed particular bacteria electricity directly, other bacteria can't use it, and those ones that can go through and use the uranium like I talked about for, or the chlorinated compounds, they can use this as their final electron acceptor. And at that part of that process, they can actually break down or transfer these dangerous compounds to stop them actually contaminating. Useful thing with this as well is that uranium contamination I was talking about, here it precipitates out. So this electrode is where the uranium will actually precipitate out, become solid. So when you want to clean up the site, you can take out your electrode, that's where all uranium is, and it's actually easy to clean up that site, so there's not a long period of contamination. But some more interesting one that's come out recently as well is that the bacteria not only can use a big wide range of electron acceptors, one of the ones they can use is CO2. So people are familiar that plants use sunlight and CO2 to grow and make carbon and make all their products. These bacteria can use electricity and CO2 to grow and produce a wide range of their own different products. So at the moment, they can actually start producing things like acetate or butanol. The hope is in the future that what we'll be able to do is use commodity chemicals. And the great thing with this is a country like Australia has bountiful amount of sunlight. Only problem is in the areas where it will be really good to have solar farms is that there's no one actually living there. And transferring electricity over those long distances is really quite difficult and you lose that power. Whereas if you could get the bacteria there to take the electricity, turn it into actually something useful like butanol or a power, something that we can use as an energy source, we can actually transfer that back, put into our systems and use that for a long time. It's been really interesting to us as well that the bacteria have been able to do this because when you look at the environment, an electrode, a cathode and anode are very quite unnatural systems. So the ability that you can find bacteria everywhere doing this actually was quite surprising, but looking into some of our research and what we've been working on is how did this evolve, where did this develop from? And what this has actually come from is bacteria are able to actually do work together. Bacteria quite often don't exist by themselves. They'll actually work in groups to break down food and they feed each other. So even though sometimes students hate it, bacteria actually go through and do work with groups, work well together, and they can do this by actually transferring electrons between each other. So one bacteria will start eating something, it'll actually transfer electrons to another one who use that energy to go do another process, and by working together they can do this. One of the big important ones can be that in the rumen, something like cows, this is how we get a lot of methane. The bacteria will actually break down, you'll break down a whole bunch of compounds, you'll get to these end products and these bacteria here. Some can use volatile fatty acids, feed them to methanogens, they produce methane. Only problem for us is we get a greenhouse gas and our cows will burp and other things a lot. But this is a natural process. But by understanding this natural process, we can look at these mixed communities, we can look at these ones together at how all this type of electron transfer is able to occur to actually go and try and mould their actions, trying to actually push their behaviour in certain ways, to actually look and see how we can actually manipulate it. Because as well in my lab, as well as the electron microbiology, we also look at plant and microbes and animal and microbe interactions. And the interesting thing can be is that this electric microbiology does have this implications in this animal microbe interactions. I have some work where we're looking at a mouse model of autism as well. And what we've been looking at is how the actual brain interacts with your gut. Your stomach is full of bacteria, and your stomach actually communicates back and forth with your brain. So bacteria can be partially responsible for modulation of your mood and behavior, but your brain also can affect the stomach, the bacteria in your stomach. And with this mouse model for autism, we have a mouse model where there's a single nucleotide mutation in a protein in the brain. And that single nucleotide mutation not only changes the mouse behavior, but actually changes the bacteria that's present in their stomach. So the work we're interested in looking at now is how that bacteria as a community gets changed, how that change affects behavior, and can we actually go through and help reverse some of that? So it's not going to be a cure for autism, but if you can reduce aggression, if you can reduce anxiety, if you could actually help modulate these behaviours, then you can actually help overcome some of these sort of things that are impinging on the person's life. And similar not only in that, but also there's a range of different other ones. There's genetic variations, there's things like different drugs that have different effects on these microbial communities. But the thing that we've actually had some success in is that when you're actually looking at something like a calf, if you can understand how these communities work, you can actually have dietary intervention, you can feed the right set of that bacterial community. All this one is showing is that what we've done here, by feeding the cows the right things, we've changed this electric interaction here. So we're getting less of these methane producers, we're getting less greenhouse gas, so overall we're getting actually healthier cows and it's better for the environment. So this is not going in and changing things other than by changing what is getting fed to what. So this is sort of how I explain a lot of the work, but this then is how marketing explains the work as well.
Bacteria has a bad reputation, but really only 1% are dangerous. Worrying about only bad bacteria can make us lose sight of some of the important things. It's like using a flamethrower to weed the garden. Did you know some bacteria can be electric? They can eat electricity. They can breathe electricity. And they can pass it around by touch. And when they work together, we can harness their electric power to perform for us. All these bacteria are helpful because of all the useful things they can do. Some bacteria use their electric skills to turn waste into things we can use. They can even produce enough electricity to charge small batteries. Now that's helpful bacteria. Helpful bacteria could also improve our health. Bacteria in our gut affects our brain. When it gets out of balance, we can feel anxious or depressed. Bacteria are very much a part of us. Helpful bacteria could restore balance in the gut, keeping our bodies and brains happy. That's really helpful bacteria. When bacteria play together, they can generate electric change. Helpful bacteria. This is what we're working on.
What? Well, I'm not sure if that was because marketing my suggestions, how they took them or not, but my wife holds a record of 3 minutes and about 12 seconds of laughing at that one at the end. But just that today overview, what I hope I can actually convince to you is that bacteria can produce electricity, they can eat electricity, they can actually feed electricity to each other. So we like to think we're all very modern, we all think we're very evolved. Bacteria have already had electrical grids for a long time. But understanding how this occurs and the driving factors and the important inputs will allow us to bioengineer natural communities for beneficial outcomes without actually working out how to push these ones in the right way to get them to do what we want, so we can actually affect things like the environment, our own gut, mood, lots of these different things come into play. But all this work is not my own. I'm lucky to have a very good large lab. A lot of people are doing a lot of this work and doing all the nitty-gritty. Also a lot of collaborators around the world and also throughout Australia. So with that, I'll say thank you and happy to answer any questions if anyone's got any.
Thanks, Ashley. That was awesome. I'm like, you can clean up uranium. I want you to clean my house while I'm are there any questions for Ashley?
particularly good at setting jobs like this?
Yeah, so we've got work with people like, one of the companies we're working with is Western Water. They have a problem where the bacteria will breathe sulfur. Thing with sulfur, it makes hydrogen sulfide. That will actually dissolve your pipes. And if you've ever have a sewage pipe that's been dissolved outside your house, you know that's a big problem. But what we're doing there is looking at those to using the electric bacteria instead of breathing sulfur to breathe the electrode, take the electrons away. Depending on the courses, what is happening, what is the energy, what is the actual sort of community. And that's why we talk about community drivers and community function, because if there's a function that's happening that we can actually go through and change, that helps us. Same with the methane production.
Any other questions? All right, well, please join with me in thanking Ashley and Cheryl again for two amazing talks.